Okay, hello everyone, and welcome to the GWAS seminar series. Can I just get an indication if everyone can hear me? Perfect. Okay, so good afternoon and welcome to the sixth lecture in the HCA Bionet Genotyping Chip Data Analysis and GWAS lecture series. And in this lecture, we'll be introducing the fundamentals of genotype imputation and its importance in GWAS. And the lecture will provide an overview of reference panels, file formats, algorithms, and tools used for imputation. But as usual, before we start, just a few house rules. So for everyone that's logged in, we ask you to please make sure that your mic is on mute throughout the entire talk, so we don't hear any background interference. If you do need to activate your audio, it can be activated by clicking on the mic icon on the top left-hand corner in your browser. If you do have a question during the lecture, feel free to type it into the chat box. Also, you can use there is an icon on the top panel to raise your hand if you do have a question. But we encourage you to please type in your questions into the chat, and then we'll get to that during the Q&A session. And also just to mention that all previous lectures are now available on the HCA Bionet YouTube channel, and I've put the link in the notes box on the bottom left-hand corner of the screen. And then also very important, at the end of the lecture, again, once the lecture is completed, you'll be redirected to a URL to complete your attendance form. So please make sure that you do that, especially for those who are attending the hands-on workshop. Okay, so moving on in our GWAS series, we are very excited and privileged to have Dr. Dhriti Sengupta with us today to present today's lecture on imputation and GWAS. So Dhriti is a postdoctoral fellow at the Sydney Brenner Institute for the Molecular Bioscience at the University of Witwatersrand in Johannesburg, South Africa. She received her PhD in bioinformatics from the University of Kolkata, India in 2013. And her PhD research involved applications of system approaches to studying protein architecture and gene regulatory networks. Her research interests include studying population structure and admixture, informing demographic history, as well as identifying signals of selection in the genome. She's currently conducting the imputation for the AwiGen GWAS dataset, and is also an analyst in the AwiGen population structure study and the HCA whole genome sequencing study as well. So with that, over to you, Dhriti. We're looking forward to an interesting lecture.
Thanks, Sean. Can you hear me?
We can. You might speak up a little bit louder.
Okay. So good afternoon, everyone, and thanks for being here at the lecture. So as you know, today's lecture is aimed at introducing what is imputation and why it has become a routine in association studies, and also the nuances involved. So to begin with, I will discuss the concept behind imputation, then we will discuss the current reference panels and tools used for imputation and the factors affecting imputation quality. Though these things can get very mathematical, I have minimized that part, as it will be difficult to explain everything in an hour. I will try to make it as informative as possible, hopefully without overwhelming you. So my attempt is mainly to introduce the concept and then a dry run on how you would be imputing your own genotype data, and most importantly, how to assess the quality of the imputation. So moving on to the first slide. Statistically, imputation is the process of replacing missing data with substituted values, right? And imputation finds its application in epidemiological studies, clinical studies, and many other areas. Wherever you have to deal with missing data, imputation is useful. In case of genetic studies, based on a set of available genotypes, as shown in the figure on the left bottom, you try to get as many as possible genotypes using this technique. As you can see, the gap in the initial genotype data shown in the question marks in the bottom left figure has been filled with genotypes after imputation in the bottom right figure. The top plot shows the p-values for associations before and after imputation. As you can see, the major utility of imputation is that some of the main signals missed in the initial data perhaps the particular SNP was not genotype are identified in the imputed data set. So let's now revisit the history of this science, summarized nicely in a recent review by Shayantan Das in 2018. So the first ever GWAS study was done back in 2002 by Tanaka and group, and they studied genetic susceptibility of myocardial infarctions. And soon after that, there were many successful stories in the field of GWAS. One of the landmark studies was done by Klein and group in 2005, where they identified regulatory genes in inflammatory pathways associated to macular degeneration. This study was important because for the first time they used the HapMap as a reference panel. Another major success in GWAS history came in 2007 when Wellcome Trust Case Control Consortium launched the largest GWAS study of that time that analyzed 17,000 cases and controls for several common diseases like coronary heart disease, Crohn's disease, rheumatoid arthritis, type 1 diabetes, and so on. And they were very successful in identifying multiple risk loci for several of the diseases, including Crohn's disease and rheumatoid arthritis. So this led to what some scientists say the genomic gold rush, and ever since then a plethora of association studies on common diseases have been carried out with different degrees of success. One of the sources for the limited success was suggested to be the limitation in SNP genotyping arrays, as they only a small fraction of the human genetic variation. And this is where genotype imputation jumped in, a smart and cost-efficient way of analyzing rare variants in GWAS studies. Variants, as you have seen in the previous slide, although not directly assayed on the GWAS array, can be imputed using genetic information from a reference panel. In a real-world scenario, after starting from around 2 million SNPs, you can easily get up to 15 to 20 million SNPs after imputation. So before we go into details, let's continue with some more history. So one of the first studies to use genotype imputation was on the type 2 diabetes in Finnish samples done by Scott and colleagues in 2007. Imputation helped them identify and replicate multiple risk variants and compare their results with other studies that use different genotyping arrays. After another study carried out by Wellcome Trust Case Control Consortium was also published in the same year, where they looked more on the evaluation of the imputation as a whole. Since then, imputation has been a key step in GWAS studies, as it finds its application not only in increasing power of the association studies, but also improved fine mapping efforts, and importantly, is very essential for combining results across the studies. There is a great review by Jonathan McNee back in 2010 discussing these things in great details. And like I said, imputation finds its application in many ways. I will discuss them in the next slide. The first one is fine mapping. It provides a high resolution view of genetic regions by adding more variants and thus increasing the chances of identifying a causal variant. A study on blood triglyceride levels that aimed to fine map the GCKR gene found that the strongest signal came from a missense variant that was imputed. Similarly, the WTCCC data found the strongest signal in a type 2 diabetes gene coming from a gene like TCF7 coming from a variant of a genotype variant. Another essential application of genotype in imputation is meta-analysis. It was difficult to combine results from a same trait but different studies, as some of the genotyping chips used assays that the samples do not have same variants or variant correlated with this variant, which makes it difficult to merge. For example, only 20% of the SNPs included on the Affymetrix 1 million SNP are included on the Illumina 1 million array. Imputation allows the combination of results across studies. By imputing the multiple datasets against common reference panel, one can generate a common set of variants which can be analyzed across all studies to boost power. This approach also helps in identifying several new loci for different traits such as type 1 diabetes, psoriasis, and many others. Imputation in cases can also increase the power to detect an association signal. By definition, the power of any test of statistical significance is defined as the probability that will reject a false null hypothesis. That is, in a GWAS, it is the probability that a study will be able to find an association when there is one. So this is especially when there is a missingness in your data. I hope you remember what is missingness, as Sean you heard in Sean's lecture. Sometimes when SNPs are only genotyped in a proportion of the samples, imputation can increase the effective sample size by filling in such missing samples. Some simulation studies have shown that imputation can in fact increase power by up to 10% when compared to testing only the genotyping SNPs. However, some other studies have predicted a more modest gain in the power. So here are two plots showing the success stories of imputation. The plots here are similar to local zoom plots and illustrates association of genetic variants around a selected risk gene. As you can see, y-axis here represents the strength of association at each SNP, measured as -log10 p-value. The x-axis is representing the chromosomal location for each SNP. Results are directly genotype SNPs. Results for directly genotype SNPs are shown in red, and the same for the imputed SNPs are shown in blue. The figure on left illustrates a study on triglycerides and cholesterol in Sardinian populations, where a common variant in a known risk gene, LDLR, was missed when the genotype SNPs were analyzed, as it was not well tagged by any of the variants in the Affymetrix genotyping arrays. However, the SNP was identified once the imputation was performed. The figure on the right side demonstrates another example with the G6PD activity, where although there was evidence for association in the region prior to imputation, the signal increased substantially after imputation and reached the genome significance level. There are also some other benefits of imputation, like correction for genotyping errors and imputing non-SNP variants like copy number variations. So undoubtedly, high coverage sequencing is the most accurate option for these purposes. But if you compare the cost of $1,000 per genome for sequencing versus say $100 per genome of SNP arrays, and given that imputation costs nothing, we get a fair deal, right? So I hope so far I have been successful in explaining you why imputation is such an important step in the association studies. Now let's move on to how genotype imputation actually works. So estimation of missing data, as you can understand, is always very challenging. It's even more challenging when imputing SNPs arrays because of its high missingness. We know only 0.1% of the genome is typed. Moreover, you have to deal with like recombination, linkage disequilibrium, mutations, and so on. So you can imagine there was a need for highly specialized tool for this. The basic ingredient for doing your imputation analysis will be, of course, your GWAS data that needs to be imputed, imputation tool, and most importantly, a reference panel of densely sequenced genomes. Now, these reference panels don't have missing data and are used to provide allelic patterns of the population. The imputation tool directly matches the short segments between the study sample and the reference panel. This needs a bit of more explanation, right? So, individuals with similar ancestry share short stretches of chromosomal segments. We call this sharing to be due to identity by descent. And very crudely speaking, the main objective of the imputation tool like Impute2, Minimac2, and Beagle is to identify such regions of the chromosomes shared between the study sample and the individuals in reference panel. These tools model the regions as mosaics of haplotypes in the reference panel. So what you are seeing in the left of the panel here is the schematics of the same. Now, for many imputation tools, as you saw in the first slide, have been developed by different researchers over the last decade. Most contemporary imputation tools employ a hidden Markov model framework. Though explaining this in details is out of the scope of this lecture, but in short, these tools assume the observed genotypes of the study samples with missing data as the observed states of HMM, and the set of reference haplotypes represent the hidden states of HMM. These tools differ in how they define the state space and the parameters of HMM. Imputation is very computationally intensive process because of the probabilistic nature and a high missing rate of the data, right? And an important step to reduce the burden was the introduction of pre-phasing. So the first step in the process of imputation nowadays is pre-phasing your data, where you estimate the haplotypes of the from the GWAS genotypes and then impute the phase data. Just to explain what's going on in this step, the figure on the right side shows genotypes of an individual. We know genotypes are actually another combination of alleles at each site. As you don't know which one comes from the mother side and which one comes from the father side, you can have many probable phases. And there are tools like Eagle2 and Shape2 which try to reconstruct the correct and most probable haplotypes here. So while earlier tools did phasing internally as the part of the imputation, now it's done separately, and this has reduced the computational load almost quadruple times. The quality of imputation has improved greatly with time. Though improved genotyping technologies and more efficient analytical methods are some of the factors, the major driving force has been the development of NGS technology that led to more individuals being sequenced for large-scale studies like 1000 Genomes Project and UK10K. The reference panels we had earlier days of imputation were based on about 1000 individuals and had 1 million SNPs. So the number of SNPs imputed were limited. But as sequencing is getting better and cheaper than before, there is a rapid increase in the sizes of the data sets used as reference panels for imputation. For example, we have now cosmopolitan reference panels like the 1000 Genomes Phase 3 that includes almost 80 million SNPs and 2,500 individuals. This is one of the most commonly used reference panels, as this is also publicly available for downloading. Then there are some even bigger but mostly Eurocentric haplotype reference panels like the Haplotype Reference Consortium panel, which has about 30,000 individuals. However, the HRC along with another large reference panel, TopMed, have some restricted access, and it's only partially available for publicly downloading. The reason researchers are aiming for larger reference panel is because with larger reference panels, you have an increased catalog of genetic variants leading to a better imputation accuracy, which will finally lead to improved power of downstream association analysis, right? And here are some of the commonly used tools for imputations. The ones shown in the red box, including Deagle, Impute2, Minimac3, are the commonly used imputation tools so far. You can see a sharp rise in the number of tools developed in the last 3-4 years. This is because the size of the reference panel increased. Like as the reference panel sizes increased, there was need for more and more computationally efficient algorithms. Like one of them is prephasing. In today's lecture, we will mainly talk about Imputed2 and Minimac3. So this brings me to the end of the first part, where we discussed about the basic concepts of imputation, the tools and the reference panels needed for imputation. So, any questions? Okay. So let's say you have a GWAS data with you and you want to impute the data. How would you proceed with that? So the first step would definitely, like any other genetic study, will be to clean the data. So you have to convert your data to PLINK or VCF format and remove all samples with excessive missingness, sex mismatch, exceptionally high or low heterozygosity, remove outliers in PCA and duplicate samples. Similarly, remove the SNPs with high missingness, excessive low mass, deviations from Hardy-Weinberg equilibriums, and remove the duplicate SNPs also. From Sean's lecture, you already know why you need to do the QCs and how would you do them using PLINK. There's just one additional point. If you have multi-allelic SNPs, which might come from merging datasets generated on independent genotyping arrays, you should decompose them first. That is, you have to split them by allelic variants. We don't remove related individuals from this data. This is because having related samples actually helps in your imputation. Continuing with the data cleaning, the genotype data should be in genome build 37, or rather it should be in the same genome build of the the reference panel is. In case the build is different, you can use the UCAC leftover software to perform a genome scale conversion from one genome build to another. Also, please make sure that the reference allele in your data is matching to the genome build 37. This is important because at times PLINK causes major and minor allele swap that can lead to information loss. So if you're not sure, please use PLINK command like the A2 allele or the that's another command, that is real ref allele, to set reference alleles. And just for information, you can use PLINK's command keep allele order to prevent the major minor swap. And last but not the least, it is absolutely essential to align your genotypes to the same strand convention as the reference panel. This is non-trivial because genotype data from Illumina are essentially in their specific top-bottom format, whereas common publicly available reference panels are aligned to the plus strand. Therefore, SNPs aligned to different strands may have different alleles. For example, like if you have A or AG in your dataset, while it is GC in the reference panel. So any SNPs whose strand issue cannot be resolved have to be removed. However, do not worry much, as they can always be imputed back to your data. There is very useful site maintained at Oxford, which contains all the strand orientation information, reference alleles, and position of variants for all the commonly used genotyping chips. And the good thing is that they have the information in all AG 36, 37, and 38 genome build. This site is really helpful if you are stuck at some of the commonly faced problems like the ones I mentioned above. You can take the information from this site and use PLINK commands to prepare your data. And just in case you are wondering if our H3 chip is there, not yet, but we are in touch with them and hopefully it will soon be there along with other arrays. Okay, so once you have cleaned your data, you have to phase it to get the best guess haplotypes, right? We use tools like Eagle2 and Shape2 for phasing. Some programs are designed to run on small segments of the chromosome. You merge them later, and other tools can run on whole chromosomes. There is also debate on the best performing tool for phasing. Some researchers consider Eagle2 to be the best phasing tool, while others consider Shape2 to performing better. The jury is still out on this, but in terms of ease, I prefer Eagle2. Phasing can be done with or without reference panels. If your data set is small, it is recommended to phase using reference panel. So this is the command to phase using Eagle. The -VCF ref specifies the reference panel you are using, here 1000 genomes. The VCF target specifies your data in VCF format. I will talk about VCF files later. The Chrome, it specifies the chromosome you want to phase. Like here we are phasing chromosome 20, and genetic map file needs an it needs an additional file for with the genetic map information in St. Morgan's. And finally, out prefix. Here you would specify the output file name. So once you have phased your data, you are ready for imputation. You have to select an imputation tool and reference panel against which you will impute your data. The pie chart here shows the most popularly used imputation tools. As you can see, the most popular ones are Impute and Minimac. Earlier people did use Plink for imputation, however, it's not used anymore. In this lecture, I will mainly talk about Minimac 3 and Impute 2. Each tool has its own input and output format. For example, Minimac 3 uses VCF files, while Impute 2 accepts .hap and .hap and .gen formats. Now, each of these tools have panels in their compatible formats, and the publicly available imputation panels can be downloaded from the links provided by the respective websites. The reference panel for Minimac 3 are generally in VCF or M3VCF format. M3VCF is very similar to VCF, however, can handle very large references with low memory. M3VCF is very specific to Minimac 3. Impute 2, on the other hand, provides its own scripts to convert phased VCF into reference panel format, one legend file and one haplotype file. So let me quickly introduce the file formats that you need to impute using Minimac 3 and Impute 2. VCF file stands for variant call format file. A typical VCF file, as shown at the top, would have three parts. A header, shown in the blue box, starts with hash and provides metadata describing the body of the file. Then there is an info part, shown in red box, that describes information like the name and position of the SNP, the reference allele, the alternate allele, and so on. And then there are genotypes, as shown in the green box, for each individual for each SNP. The alleles are coded as 0 and 1, depending whether it's reference or alternate, and you have to look at the info section to know what the exact alleles are. The two other formats you need to know are the HAP and the Geno formats that are being used by Impute 2. The Geno files store data in one line per SNP format. For each SNP, the first 5 columns would represent SNP, the SNP ID, the RSID, the base pair position of the SNP, and the alleles, let's say A and B. Then the next three number of the lines provide the probabilities of the three genotypes AA, AB, and BB at the SNP for the first individual. The next three should be the genotype probabilities for the second individual. This information here is unphased, and the HAP file stores the same information in phased format. The toy example here shows haplotypes for the 5 individuals at 5 SNPs. That is, you have one haplotype per column. The HAP file comes with a legend file, and that has the information for each SNP, like the RSID, the position, and the alleles. Please note, you can convert between Impute 2 and VCF files using QC tools. So now you have decided your imputation tool, you have downloaded the corresponding reference panels, and you have the phase data ready to be converted. You have, in fact, you have the phase data and you have converted into your input format, right? So finally, let's impute. The command for minimax3 is shown in top. The ref hap is to specify the reference panel. It's HRC in this example. Minus hap points to the phase data that you have just generated. The format specifies the format you want the imputed information in, and prefix is the output file name for your imputed data. The command used for impute2, as shown in the box below, is similar. There are a couple of new terms like the int, which actually asks impute to analyze the physical positions from 1 to 5 million. And you need a lot of patience, as each of these steps can take a lot of time to run. So now most imputation tools provide you with genotype probabilities and allelic dosages. So what is genotype probability? In the figure on the left, you can see that the third position is uncertain in the sample genotypes. However, in the reference panel, 3 of the 4 possible genotypes are CC and just 1 is CT. The probabilities of our examples at the third position would be for CC is 0.75 and for CT it's 0.25 and TT is 0. The probability is 0. This is just a super simplified example, as in real life you need tools mentioned above to do this, calculate these probabilities. The second thing provided generally is allelic dosages. Allelic dosage is the probability of observing alternate alleles and follows the formula shown in the top right. Here, based on the genotyping probabilities, considering C as the alternate allele, the allelic dosage is 1.75. You just have to calculate it using the genotype probabilities. The allelic dosage ranges from 0 to 2. This value closer to 0 would indicate homozygous differences, 1 heterozygous, and value closer to 2 indicate homozygous alternatives. The advantage of allelic dosage over genotype probability is that it decomposes the 3 values into 1 and also enables us to assess the strength of the alternate reference allele. The gold standard for assessing imputation quality is to compare the true genotype data with the genotype probabilities. However, that's rare to find. Therefore, we can use certain measures based on the GP and the allelic dosages to do this. Please note that the allelic dosage and genotype probabilities are calculated per individual to provide uncertainty over that genotype. To assess the quality of imputation for whole dataset, you need to use R-square, a computational derived correlation between the true and imputed allelic dosages. I'm not going into the derivation, but it can be estimated from the posterior allele probabilities without knowing the true alleles on each chromosome. So, Minimax generates 2 output files. The first output file, which is the .info file, looks like this table. The file begins with the identifier followed by the reference and alternate alleles and then frequencies of the alternate and minor alleles. As you might know that though there is a very high correlation between alternate and minor alleles, they are 2 different things. At times, the reference allele can be minor. The definition of minor allele might be different in African populations and European populations. So moving on to the highlighted column, we have mentioned R-square just now, right? This is basically the confidence in the imputed dosage. It varies between 0 and 1. The higher value close to 1 indicates good imputation, while a value closer to 0 would indicate poor imputation. For example, the SNP shown in green has the R-square of 0.8, whereas SNP shown in red has R-square value of it's low, right? So as we see, the low R square values SNPs, so the SNPs with low R square values are subsequently removed in downstream QCs. There are many values provided in Minimax 3, but we are not going to discuss them today. The second file produced by Minimax is the VCF file. It looks like any typical VCF file, but if you look at it carefully, you will find some additional information not contained in a sequence-based VCF file, like the one I showed you just a while ago. The top box shows the header, the middle box shows the SNP information. So here you can see the R square values are included in the info field. If you go at the sample levels, I just zoomed it at as another box in the bottom, as the original thing was not fitting. So you can see for each individual, this VCF contains, in addition to genotype information, that's in GT, the allelic dosages and the genotype probabilities. And if you remember, we specified this format in the command we used for Minimax 3. I would also like to mention that there is a tool called a dosage converter, which converts dosage files in VCF format from Minimax 3 to Plink and other formats. This is specifically helpful if someone has been using the previous versions of Minimax 3 and have a pipeline that's already developed for downstream analysis using Plink or Mac. Impute2 also provides genotype calls, genotype probabilities, and an imputation quality score called InfoScore that is similar to R square for Minimax. However, the derivation is slightly different. As you can see in the scatter plot here, there is a fairly high correlation between the InfoScore and the R square. However, there are some differences as well, specifically in the higher values, and that is why it is not advisable to compare against one another. Similar to R square, the info score also ranges from 0 to 1, which means complete uncertainty, and 1 means no uncertainty. That is, it's as good as a sequence or true genotype data. The closer the info score value to 1, the better is the imputation. Another way of looking at it is that the R square or the info score for say a sample of 1000 samples is 0.8, and it implies that the amount of the data, the SNP, is equivalent to a set of perfectly observed genotype in a sample of 800 individuals. Please note that the allelic dosage information is missing in Impute2, and you need to calculate that. So now we come to imputation evaluation. So imputation performance is evaluated on the basis of 4 main parameters. The first is the number of SNPs imputed. Just to give you an estimate, if you start from a set of 1.8 million SNPs corresponding to a 2.5 million array, you can go up to 32 million SNPs in a sample size of 4,000 individuals. The second parameter is the number of SNPs in the MAF ranges. This provides you estimates of how many rare and common alleles have been imputed. The third parameter is the number of SNPs with good imputation scores. The green box shows you a command to extract SNPs based on R-square cutoff in BCF tools. After running this command, you will be left with only well-imputed SNPs in your dataset. The fourth parameter is the relation between imputation quality and the non-reference allele frequency. The plot on the right shows aggregate R-square per allele frequency bin. To calculate the aggregate R-square, you have to first separate the SNPs at different allele frequency bins and then calculate the average R-square for the SNPs at each frequency bin. So in this figure, the four reference panels have been compared. Irrespective of which reference panel you use, imputation, you can see, is most difficult for rare variants and gets better with increasing non-reference allele frequency. We will come to comparison of panels in few slides. To study the imputation qualities across the genome, a common method is to plot the R-square along chromosomes. The three plots on the left shows three different levels of imputation. Please note that the axis is the x-axis is the genomic coordinate and the y-axis is the R-square ranging from 0 to 1. The plot, the top of the three plots shows the worst imputation. This can be inferred from the high density of dots across all R-square values. The second plot is slightly better and shows higher frequencies closer to 1, but there are still dots all across. The third plot is what you would like to see, high density dots around 1, implying good overall imputation of the data. The plot on the right shows the same data, some as frequencies of histograms, with x-axis showing R-square values and y-axis showing the frequencies of imputed SNPs, and they tell you the same story. Another way of looking at imputation quality is to study the correlation between minor allele frequencies of the SNPs in the imputed data and the reference panel. As you can see in the plot, the left one shows no correlation at all, implying a poor imputation. The plot on the right shows a very correlated correlation between the allele frequencies of the imputation panel and your data set, ensuring the imputation has been good. However, in these days of multi-ethnic reference panels, it is important to choose the reference panels that is genetically or geographically closer to your population, as the same bad plot can also be arrived if you have a decent imputation data but discordant reference panels. In addition to these data, there are method-specific parameters which also provide a good estimate of imputation quality. This is a table called concordance table produced by the Impute2. The concordance table actually shows the results in an internal cross-validation where Impute2 performs, it marks the genotype of each variant at a time in the study data, then imputes the masked genotype with information from the reference data and nearby variants. The imputed genotype are then compared to the original genotype to evaluate the quality of the imputation. The sample here you can see it's well described already. This estimate also gives you an idea about how well your data has been imputed. So while we are evaluating the imputation quality using existing tools, you also might wonder which performs better in terms of efficiency and accuracy. So here is a comparative study on some of the presently used imputation tools. What we see here is that Minimax 3 consistently outperformed all the alternatives, both in terms of memory needed for imputation and irrespective of the reference panel size. For example, it needs less time to impute both KGP and HRC reference panels. The imputation quality across the four methods was also compared by calculating the squared correlation coefficient between the imputed allele dosages and the masked genotypes at different allele frequency bins. Surprisingly, if you look at the mean R-square values for the different methods, you will see these methods are almost similar in terms of imputation accuracy, which is very reassuring, right? So far, we saw the usage of different tools, even though they have difference in terms of computational efficiency. It does not affect imputation accuracy much. So what are the other factors that can actually affect imputation? Of course, size and sequence coverage of the reference panel. That is the first main point. The other factors are the genetic similarities between the reference panel and the study samples. I will show you an example in the next slide about this. The next point is minor allele frequency of the variant to be imputed in the reference panel. This is because if the frequency is low, the probability of it being imputed is also very low. The density of the genotype also matters. The denser genotype array increases the number of sites to match with, thereby improving the chances of finding a shared haplotype segment. So the plot in this slide will answer to questions like why we need so many reference panels and how genetic similarity between reference panels and study samples can affect imputation. The plots presented here actually illustrates the imputation accuracy for 5 ancestries: European, American, East Asian, South Asian, and African. How they performed the analysis is they extracted 10 samples from each of these ancestries from 1000 genome phase 3 data and downsized them using the Illumina 1 million SNP chip array and imputed them using the different reference panels like TopMed, haplotype reference consortium, and the 1000 genome phase 3. So the aggregate R square is plotted as a function of alternate allele frequencies. What these figures unanimously demonstrate is that we see the imputation accuracy always increases with the minor allele frequencies. You can also see that for European ancestry populations, TopMed and HRC, both predominantly European, are almost performing similar. TopMed imputed a low frequency better than HRC as it is a reference panel made of high coverage sequence data, which can impute rare alleles better than the low coverage reference panels. However, if we see the East Asian and the South Asian ancestries, we see 1000 genomes imputing mildly better than TopMed or HRC, as none of these population had significant East Asian or South Asian samples in the reference panels. So even though HRC is way larger in size than 1000 genomes, the former imputes better as they have the representative populations in the panel. And finally, for the African ancestry population, we see TopMed outperforming HRC. This is likely because TopMed panel was generated, as I mentioned, high coverage sequence data, and hence it had more alleles to be imputed against it. So, although there are quite a few reference panels in the public domain, given the diversity of populations and scarcity of whole genomes from most geographic regions in the current imputation panels, the search for the ideal reference panel is still on. Therefore, if you are imputing a European data, you have probably the best panels already there. However, if your samples come from South Asia, East Asia, or Africa, the best reference panel is not obvious. The paper in the center middle shows that in spite of huge size, HRC panel still performs inferior to 1000 genomes in African populations, similar to what we saw in the previous slide. The paper in the bottom right shows the same thing for Chinese populations. Another avenue for improving imputation is that people are trying to a lot these days to combine the existing reference panels as discussed in the paper shown in the top right. This can be done either by combining the reference panels before imputation or by merging the results from independent panels after imputation. The Venn diagram on the bottom left from the paper shown above the plot illustrates this nicely for an African population. This paper also reiterates the necessity of an integrated reference panel for Africa. You would also be glad to know that H3 Barnett led by Eaton and Nicky are actually trying to generate an African specific reference panel using high coverage novel sequence data. So we are going to have our own African reference panels. And just to give you a flavor of how reference panels are made, I am showing you a rough schema based on the HRC panel construction. To begin with, they took VCF files for each dataset, one chromosome at a time. For each VCF, they generated a list of sites that were called on it. Now these sites are merged to generate a combined site list. This list was then filtered using minor allele count cutoff of 5. Now this merged list was used to conduct genotype calling on the BAM files, one sample, one chromosome at a time. The BCF files generated after genotype calling were then merged. Then two-step filtering process, first filtering the sites based on genotype quality, MAC, Hardy-Weinberg equilibrium, etc. was performed. This was followed by removal of individuals. The individuals removed were either duplicate or individuals with low genotyping rate. This gave the final reference panel containing about 45 million SNPs with an overall MAC greater than 5. So if you're already terrified with all the steps you have to go through to impute your data, there is a good news for you. There are two online imputation servers that almost do everything for you. The Michigan Imputation Server and the Sanger Imputation Server. Both of them require, they are kind of very popular among researchers, and all you have to do is to upload your cleaned data to their server, either using secure file transfer protocol or Globus. You need to have an account in Globus for that. There are two Globus endpoints, one being your laptop or your cluster, wherever your genotype data is stored, and the other endpoint being the imputation server. The imputation server does the pre-phasing and imputation for you and stores your data in a zip format. You are notified via email once the job is finished, and you just download your data. It's very simple, right? And other than making you lazy, the other reason why it's probably best to use these imputation servers is because of the reference panels. Like I mentioned previously, there are some reference panels like HRC, which is only partially available for downloading. However, you can have the access of the full reference panel via these servers. So this is the home page for Michigan Imputation Server, and you can see on the top right corner that so far it has imputed about 18 million genomes and have 3,000 users. You can choose your own pipeline, like the reference panels and the phasing tool from the drop-down options provided. However, Michigan server only allows you to use Minimax 3 for imputation. And this is the second imputation service provider from Sanger. Similar to Michigan server, this one also allows you to choose the reference panel and the phasing tool. Sanger, however, uses a PBW2 algorithm for imputation. This is one of the only tools which do not implement HMM and instead use the post-Borrow-Wheeler transformation to find the set maximal matches at each marker of the genotype sample, which in turn is used to assign alleles to the missing markers. Just a brief walkthrough how you do it. Both of these servers need the input data in VCF format, while Sanger imputation server needs a single large VCF including all chromosomes as input. The Michigan imputation server needs them chromosome-wise. The good thing is that unlike some other softwares, these imputation servers are very well documented. However, the Sanger imputation server assumes that your data is already QC'd. So you need to QC your data very stringently, otherwise it will just abort the job throwing you errors. And you do it similar to the way I showed you previously, like matching, strand matching and reference allele matching. A Michigan imputation server, on the other hand, does a thorough QC before pre-phasing your data. The rest of the procedure is same. The server imputes your data, and after you have downloaded your data, the server deletes the data in few weeks. They claim to only store the general statistics like a number of imputed and phase samples and markers, and I do believe them. And so by now you understand how to read this plot. Here 10 individuals of European ancestries are imputed against HRC reference panel and Michigan imputant and in Michigan server. So this one is mainly to compare the how the reference different imputation servers perform. So, and you can see here, Michigan server shown in red imputes slightly better than the Sanger imputation server shown in black. So this is where I end my presentation here. And so these are the take-home messages. Imputation is a powerful method to improve the outcome of GWAS study by making a number of additional analyses possible or simpler. We have discussed parameters such as genotype probabilities, allelic dosages, imputation quality scores like info scores and R squares to evaluate the imputation quality at SNP level. Parameters such as number of variants imputed, number of high quality variants imputed, genomic distribution of the imputation quality scores, allele frequency comparisons provide an estimate at the dataset level. Factors such as sizes and sequencing coverage of the reference panel, genetic similarity between reference panels and study samples, density of the genotyping array affect quality of the imputation. There are various tools for imputation. We have discussed Minimax 3 and Impute 2. Although Minimax 3 performs better to Impute 2, we do have Impute 4 and Minimax 4 which claim to perform comparably. So perhaps you will have a lot more tools to choose from. What seems critical at this moment is the choice of reference panel, as in a non-European population, as we all are dealing with, it might make a huge difference. So, and finally, I would like to thank the organizers for this wonderful series. Even I'm learning a lot, and thank you all for your kind attention. Thank you and happy imputing. So, any questions?
Thanks, JT. I think we have a couple of questions. I'm trying to go back.
Okay, let's see.
In the chat. Okay, so the first one is, I'm trying to understand Fauzia's question. Maybe she can just type it in again. Miriam asks, what's the difference between imputation and genotype inference?
So, can you, I just am completely lost. Yeah, we have had fluent interconnected throughout. Sean, can you help me with
Sure.
the
So Miriam asked, that's in the chat. I'll just read it out to you. What's the difference between imputation and genotype inference? What is the difference between imputation and genotype inference?
It's unrelated. I mean, genotype inference is family-based and family-based study, and imputation is for unrelated, like individuals.
Okay. Then Oscar is asking, are countries not represented in the reference panel for HCA at a disadvantage when it comes to imputation?
So the Michigan imputation server, they have a tiny reference panels from African Americans, and so it's true we do. So if I understood correctly, Oscar, your question is the African reference panels, the African populations are not well represented in the reference panels, and would that affect the H3 imputation?
Yeah,
right.
that's it.
Okay. Yeah, so that's what I was answering. Like the Michigan imputation server, it has a tiny reference panel of African Americans, and so far the Sanger imputation server, it has a African specific reference panel, but it's not for public. It cannot be downloaded publicly. Yes, so can be used. It's there in the reference panels.
Okay, and then Manzin asked the question, how to deal with ambiguous alleles when it's more and common in most genotypes? Not sure you understand that. How to deal with ambiguous alleles.
So if there is something which I would suggest, if something is ambiguous and you're not sure of, just remove them. You can always get them back after imputation.
Okay. When Miriam was asking, will individuals with low R2 or R squared be removed?
So R square is a SNP wise. You get it SNP wise. You don't get it individual wise. So and the SNPs with low R
The
square, they need
SNPs
to
with
be
low R
removed.
squared
Yes.
will be removed, not the individuals. Okay, and then Oscar is asking, is it possible to combine data sequence at different coverage in a reference panel?
It is possible. However, for the people who are into development of reference panel, they have to be very careful about what sites they are calling and everything. So, but technically it is possible.
Okay, and then Wisdom says he's still trying to appreciate the concept of imputation. He's asking, what exactly do you mean by reference panel?
So reference panel, okay. So it's reference panel is a data set that's densely genotyped and it does not have any missing data, and you have, depending on the data you're using, like for 1000 genomes you have 2500 individuals in your reference panel. It's basically from sequence data. So I mean the data you want to impute is the genotype data, and you want to almost convert it to sequence data, and you want to do that using a reference panel that is actually a sequence data.
Yeah, so
That's
it
what
goes
we
on
are
to say,
saying.
do we have CNV or structure variant panels?
So 1000 genomes, and they actually allow you to impute index, but some of the reference panels, they don't have, they can't allow you to do indel imputation, but some of them they do have.
Okay.
So, and I saw there's a question, is there a limit to upload or download from online imputation server? So no, because I mean, it will, if you have a huge data to impute, it will take perhaps more time, but you can upload whatever. And because when you are downloading, you are almost downloading what, 50-60 gigs of data, so there is no limit there.
And there was another one, can you provide your own reference panel to the imputation server?
So to the imputation server, no. The imputation servers, they you can only you choose from the reference panels they provide you.
Okay, I think that's all that I can see in the chat. Are there any other questions? And again, while people are typing, just to mention that these lectures will go up on the HA Bionet YouTube channel. The link is here at the bottom. Put the link at the bottom. So the other lectures are already up there if people want to have a look at it.
Shall we always refer to panels to impute? So for imputation, you do need a reference
If data
panel,
is
right?
missing.
like when you're imputing your genotype data, so I mean you are genotyping because you have a lot of information missing. So, and yes, and don't be scared even if you have a high missingness in your data, just QC it very like stringently and then try to impute, because there are chances you will get back a lot of your data.
Samir is also just making the point there that you can actually also choose to do the imputation yourself. You don't have to use the servers. It's just that the servers make it easier to do that, and they also give you access to reference panels that you don't have. So if you do have your own panel, yes, you can do it yourself, but there is the servers available for you to run the imputation. And hopefully we'll have a server within HA Bionet soon to allow us to do the imputation. Okay, are there any other questions? If not, I think thanks again, Riti, for a very, very interesting lecture. And it looks like people have learned a lot. And we will see everyone next week for the last lecture in the series. And yeah, thanks, and see everyone next week. Just stay online to finish your attendance form.